This is our cells lesson. In this lesson, we'll explore the science and engineering practices of developing and using models, planning and carrying out investigations, analyzing and interpreting data, constructing explanations and designing solutions, as well as obtaining, evaluating, and communicating information. Our cross-cutting concepts include patterns, scale, proportion, and quantity, systems and system models, and structure and function. So what are living things made of? They're made of cells. Cells are the smallest living units. They come in many shapes and sizes. Some organisms are unicellular, which means that they're made up of only one cell, while other organisms are multicellular, which means that they're made up of many cells. Think about some of the organisms that you are familiar with. Can you think of some examples that are unicellular and maybe some examples that are multicellular? Cells are also made up of smaller parts. Each part performs a particular function inside the cell, helping it to work properly. The cells that make up living organisms can look very different from one another. For example, plant cells look different than animal cells. However, some cell parts are common to every type of cell in every organism on our planet. So let's do a comparison. Animal and plant cells. Both of them have a cell membrane. Both contain cytoplasm. Recall that the cytoplasm is the jelly-like liquid that fills the cell, also termed cytosol. And the cell membrane is responsible for allowing materials in and out of the cell. Both contain mitochondria. These are the powerhouse of the cell. Both contain a nucleus. And remember, our nuclei are membrane bound in both plant cells and animal cells. We call these eukaryotic cells. All of our DNA is housed there. In plant cells, we have a large central vacuole, as well as chloroplasts and a cell wall. So what similarities and differences do you see? What are the parts of a typical animal cell? We have the cytoplasm, the mitochondria, the cell membrane, and the nucleus. What structures are found in a typical animal cell? We identified each of those, the cell membrane, the mitochondria, the nucleus, and the cytoplasm. The mitochondria is where respiration takes place. Respiration is the process that releases energy from food. The nucleus is the location of a cell's DNA. The nucleus controls the way the cell works, including how it reproduces. The cell membrane holds the cell and its contents together. The membrane also controls the movement of substances into and out of the cell. And the cytoplasm is the jelly-like material in which all the cell's organelles are found and in which most cellular processes and reactions take place. The cell is like a living factory. So how can the three main parts of a factory be compared to the parts of a cell? The production floor. A factory needs a production floor on which to assemble its goods and carry out its job. It has offices and meeting rooms. A factory needs an office and meeting room to control what goes on inside the factory. Oftentimes there is a wall or a gate. A factory must have a wall to protect the inside and a gate to control what enters and leaves the site. So what does each part do? How are the parts of a cell like a factory? So the factory part that would work like the nucleus would be the boardroom and the office. And these are the areas that control many of the things that happen inside of the factory and also the cell. The factory floor would be like the cytoplasm, where reactions can be carried out. The wall and the gates would be like the cell membrane, and this is responsible for protection or regulation. A typical plant cell. What are the parts of a typical plant cell called? Our vacuole, the chloroplasts, the cell wall, the cytoplasm, the mitochondria, the cell membrane, and, of course, the nucleus. So which structures are found in a typical plant cell? We're going to explore this further. The cell wall is a rigid external barrier made of cellulose that protects and supports a plant cell. The vacuole is a cavity that contains water and nutrients, and which allows the plant cell to take in or lose water without bursting. The nucleus is the location of the cell's DNA. The nucleus controls the way that a cell works, including how it reproduces. The cell membrane holds the cell and its contents together. This membrane also controls the movement of substances into and out of the cell. The chloroplasts are the sites of photosynthesis. They contain the green pigment chlorophyll, which absorbs the sun's energy so that plants can make food. And recall that the food that plants make is called glucose. The cytoplasm is the jelly-like material in which all the cell's organelles are found, and in which most cellular processes and reactions take place. And finally, the mitochondria. This is where respiration takes place. Respiration is the process that releases energy from food. So plants are going to produce that glucose in photosynthesis, and then they are going to take that food that they produced and break it down using the mitochondria to release energy. Comparing animal and plant cells. So let's look at the structures and whether they are found in either an animal cell or a plant cell. A nucleus is found both in an animal cell and a plant cell, whereas chloroplasts are only found in plant cells. A large central vacuole is only found in plant cells. Animal cells do have vacuoles, but they're usually really, really small. Not a large central vacuole like plant cells have. Cytoplasm exists in both plant and animal cells. Cell membranes exist in both as well. Our cells only have a membrane and no cell wall. So animal cells do not have a cell wall, whereas plant cells do. So let's identify the type of cell that each passage is describing. Number one, there are several organelles within the cytoplasm of this cell, including a nucleus and mitochondria. The outside of the cell lacks a cell wall. So this has to be an animal cell because there's no cell wall. This regularly shaped cell contains a nucleus, mitochondria, and chloroplasts. Large vacuoles contain water and nutrients. This would have to be a plant cell. So what shape are our cells? Cells are not flat. They're three-dimensional. The contents of most animal cells are very similar to each other. And the contents of most plant cells are also very similar to each other. Their shapes and sizes do differ between plant and animal cells. The 3D shape of a cell is determined by its location in the body of the organism and the job that it does. So there's many different types of cells. And what do we call the different types of cells? This cell right here has a projection that's coming out of it. This is a root hair cell. The root hairs are responsible for absorption of water or other nutrients in the ground. This long cell is a nerve cell. Guard cells exist on plants, and they allow for oxygen to be released or carbon dioxide to enter the plant. Our skin cells are responsible for protection. Palisade cells, red blood cells, and sperm cells. Let's look into each one to find out more. A red blood cell is a disc-shaped cell that has no nucleus and it carries oxygen. A guard cell is a semicircular cell which guards the tiny holes in a leaf and lets gases into a plant. Root hair cells. A root hair cell is long, thin, and absorbs water from the soil. Sperm cells. A sperm cell is the male sex cell and has a tail so it can swim to an egg cell. Nerve cells. A nerve cell is long, branched, and transmits electrical messages. Skin cells. They are thin and flat, and they aid in protection. And palisade cells, they are a regular shape, and they contain chloroplasts to trap sunlight. You can make a cell model by using some different materials. You could use a plastic bag, some type of cellulose paste, small objects to suspend in the cellulose paste, and then we would need to identify what each part represents inside the cell. True or false? Are these statements about cells true or false? Plant and animal cells contain identical cell components. That is false. Plant and animal cells have some differences. Cell membranes are found in both animal and plant cells. That's true. Mitochondria are only found in animal cells. That's false. We find mitochondria both in animal and plant cells. Cytoplasm is present in plant and animal cells. That's true. Plant cells use chloroplasts for photosynthesis. This is also true. And plant cells are not specialized for specific functions. This is false. Plants also have specialized cells for specific functions. So how do we go about collecting and preparing cells for observing them? Onion is a perfect example. Onion cells are easily viewed under a microscope and can be used to illustrate the main features of a plant cell. Stage 1, we're going to carefully slice an onion so that a piece of the membrane layer is exposed. Stage 2, using tweezers, we're going to peel away a section of the membrane and place it on a clean microscope slide. Stage 3, we're going to add a drop of methylene blue or iodine to the onion cells. This will enable you to see the cells and their organelles more clearly. Stage 4, we're going to carefully add a cover slip, making sure that we don't trap any air underneath. And then we're going to observe under a microscope. So how would we go about examining human cheek cells? Cheek cells are easily obtained and can be used to see the main features of an animal cell. We're going to take a Q-tip in stage 1, and we are going to gently scrape the inside of our cheek. We could do the same thing with a toothpick. This will transfer cheek cells to the tip. Stage 2, we're going to wipe that Q-tip or that toothpick across a clean microscope slide and transfer the cells. Stage 3, we're going to add a drop of methylene blue or iodine to the cheek cells. Again, this allows us to see the cells better and the organelles that exist in them. Stage 4, we're going to add a cover slip, making sure that we don't trap any air bubbles underneath. And then we are going to view under a microscope. Using a microscope, the invention and impact of the microscope. The development of the microscope has had a huge impact on society. Microscopes allow us to view objects that cannot be seen with the naked eye. Around the end of the 16th century, spectacle makers in the Netherlands found that putting more than one lens together made things look a lot bigger. This led to the development of a simple microscope that could magnify objects by about 20 or 30 times. In 1665, Robert Hooke used one of these simple microscopes to study a thin piece of cork. He noticed that the cork was made up of lots of little compartments. He called those compartments cells. Scientists later realized that cells were the building blocks of all living things. In the 1670s, Antoine van Leeuwenhoek developed a microscope that could magnify things up to 200 times. He observed organisms that had never been seen before. These were microbes. This important discovery allowed scientists to understand more about microbes and how they can cause disease. Microscopes are now an essential instrument in many types of scientific research. They have helped us learn a lot about how organisms work and the world around us. They've been developed, and they allow us to magnify objects well over 1,000 times now today. So let's explore some of those parts of a microscope. This, on top, is the eyepiece lens, also known as the ocular lens. Right here is the coarse focusing knob, followed by the fine adjustment knob. Our objective lenses are what can be rotated into place to allow for different magnification. Recall that the revolving nose piece allows for that to happen. We have a stage where our microscope slide is placed, and then oftentimes stage clips are allowed to secure that slide. And finally, we have a light source. Oftentimes that light source is down around the base of the microscope. Recall that whenever we carry a microscope, we must have two hands on it, one hand underneath the base, and the other one on the arm. So how should you prepare cells? How would you collect, prepare, and view cells? Step 1, we would want to collect onion or cheek cells. Step 2, we would want to transfer those cells onto a slide. Step 3, we would add methylene blue or iodine to color the cells. Then we would add a cover slip for step 4. Step 5, we would place the slide under the microscope. And step 6, we would adjust the magnification of the microscope so that then we could see what we are trying to observe. So what do some of these cells really look like under the microscope? If we're observing an onion cell, we would see something like this. The outside rigid structure would be the cell wall, and then the darkened color in the middle would be the nucleus. What about cheek cells? Cheek cells look slightly different. There's not the boxy shape. There is a nucleus that we can see pretty distinctly. The outside of this cell would be the membrane. How about blood cells? Blood cells would be circular. The exterior structure we would see being the membrane. The central part here is not a nucleus because our red blood cells do not have nuclei. Rather, it's a divot on the inside of the cell showing its shape. What about sperm cells? These cells have a head followed by a tail or a flagella used for movement. And egg cells. Egg cells have a larger membrane around the outside. An egg cell is quite a bit larger than many of the other cells that we just observed. We can also zoom in on each one of these to see the internal structures of each one. The invention and impact of the microscope. The development of the microscope has had a huge impact on society. Microscopes allow us to view objects that cannot be seen with the naked eye. Around the end of the 16th century, spectacle makers in the Netherlands found that putting more than one lens together made things look a lot bigger. This allowed us to magnify things. And then, of course, Robert Hooke came on the scene, as we discussed earlier, and he identified these individual compartments within cork. He called them cells, which led to scientists realizing that cells are the building blocks of all living things. As review, Antoine van Leeuwenhoek then was the first person to really look at microbes, specifically pond water microbes. And our microscopes today allow us to magnify things to great detail. I hope this was helpful.